So this last section talks about the cell cycle, cellular growth, and development. So we just talked about how the cell kind of produces proteins and does its normal function. If we look at the cell cycle here, we have interphase. An interphase is the time that the cell is not dividing. It's doing its normal function, so it's able to produce proteins, hormones, and do what it's supposed to be doing inside of the body. Interphase consists of 3 subphases, so we have G1, S, and G2. G1 is the normal function, so the cell's just doing what it should be doing. Now some cells slip into this G0. Those are for the non-dividing cells. Neurons don't divide. So if you have one neuron and it dies, you don't just replace it. So G0, non-dividing cells. So they're just going to have maintenance. They stay in that G0 phase for the rest of your life or for the life of that cell. But most of the cells in your body have the capability of dividing, and those cells are what we're going to focus on. So G1 is normal growth function and all sorts of stuff. But then when they go into S, so they've been triggered to start the division process. S is for synthesis. So in the S phase, that's when you're going to have that DNA replication that we just went through. So that's when you're going to make a complete copy of your genetic material. Then you're going to slip into this G2. Now once you make a complete copy of all the genetic material, that cell has to grow really, really, really large so that when it splits in half, the daughter cells, or the resulting cells of cell division, are going to be, pretty decent in size and equal in size. Following interphase, you enter into mitosis. Mitosis is cell division. And mitosis really has 4 phases. The first phase is prophase. What's going to happen in prophase is the nuclear envelope disappears. So you used to have all the genetic material inside of a nucleus. Well, if we're going to split this cell in half, we can't have it confined in the nucleus. So we get rid of that nuclear envelope. The other thing we do is we make sure that these chromosomes are nice and condensed. It's easier to move the chromosomes if they're condensed into solid chunks as opposed to free-floating chromatin that's just loosely kind of condensed. So we super condense this genetic material. The next thing that happens is metaphase. In metaphase, these spindle fibers kind of cause these chromosomes to align directly in the center of the cell. So in metaphase, the chromosomes line up or align in the center of the cell. Once they're all lined up in the center, it's easier to split them so that some chromosomes can go to one side of the cell while others go to the other side of the cell. In anaphase, these chromosomes split into sister chromatids. So here you can see the sister chromatids are going to migrate toward opposite ends of the cell. Then you have telophase. With telophase, a new nuclear envelope forms around each pile of genetic material. Now we want to make sure that we have equal numbers of chromosomes in each of the new potential daughter cells. So we make that new nuclear envelope, and that's going to ensure that these cells have equal genetic material. And that really ends mitosis. Following mitosis is cytokinesis. You have this cleavage furrow that forms here, and the cells are going to slowly form this furrow and pinch apart. So the process of cytokinesis, this is the splitting or dividing of the cytoplasm, and that follows mitosis. So if we go back here, we go through the process of mitosis, prophase, metaphase, anaphase, telophase, and then in cytokinesis it finishes up the division. After that, we're going to go into our normal growth and function. And then, if necessary, we're going to repeat it and keep going through the cell cycle. So where does cancer come from? Well, there's 2 genes that are involved in regulating or kind of preventing cancer from arising. There's proto-oncogenes, and those regulate the normal cell cycle. So under normal circumstances, these proto-oncogenes are telling your cells to divide when they should. Then there's tumor suppressor genes, and tumor suppressor genes are telling the cell to not divide when they should not be dividing. A mutation in either one of those can cause the development of increased cell division. So let's try to get a visual about what cancer really is and why people end up dying of cancer. So let's pretend that our classroom is a sweater producing factory. And our classroom's job is to produce sweaters for the entire school. So we have a cell in the body, and that cell in the body has a function for the entire multicellular organism, a person. So you know how to produce sweaters. You've been producing sweaters your whole life. So you're in there knitting your sweaters, and all of a sudden someone comes to the door and gives us directions of how to build new classrooms. So we get these directions, and immediately we have to stop what we're doing. We stop our normal function, and we have to start producing classrooms. Now, if you think about, have any of you ever actually built something like a classroom before? Probably not. And not only do you have to build it, but you're going to have to build it pretty quickly. So you're going to get some materials, and now you stop producing sweaters, and your job is to produce new classrooms. So you're building them, and you're throwing up walls, and the walls are really disarrayed, and they're not perfect, and they're not structurally sound. They actually look really weird because you've never built a classroom before. That's kind of what cancer is. So when you have this dysregulated cellular division, these cells are going to start dividing, but they divide quickly so that it's not done properly. And they don't really know what they're doing when they're dividing. They're just making lots and lots of copies. So the cells really start to look abnormal. That's going to stop the normal function of the tissue, but while you're producing all of these abnormal cells, it takes a lot of energy. I mean, can you imagine? You're used to sitting in your desk knitting sweaters, and now all of a sudden you're making a classroom an hour? I mean, you're like overworking yourself. So you're going to call for energy from other classrooms. You're going to say, hey, I need more energy in here. And that's what tumors do. They start stealing energy from the body. Tumors can also promote the growth of blood vessels into them. So now they have a direct line to your blood supply, direct unlimited nutrients. So they're going to promote their own growth and development by stealing your nutrients as a multicellular organism. Now these tumor cells can dislodge, and then they can get stuck in new parts of the organism. So one of the people in our classroom that ended up getting those directions to make new classrooms, you're going to leave and go down the hall and tell culinary arts, hey, stop producing food, we need to make new classrooms. Well, now that next part is now wasting a whole bunch of energy to produce new classrooms, which is completely unnecessary. And now someone from there goes to the next spot, and the next spot, and you can see how cancer spreads around the body. All the while telling those cells to stop performing their normal function, to start making tons and tons of copies of themselves, and to do it in a haphazard way so that you end up with these non-functional, weird-looking cells, and it's energy consuming. So when people die of cancer, yes, they can die of the organ failure that's associated with the cancer, but you usually notice cancer patients get really skinny. And that's not just from the treatment. That's because the tumor itself is using a lot of your natural resources, your energy and your nutrients that you need to survive. So cancer isn't just caused by one mutation. Recent research has found that it's actually an accumulation of mutations that happen over the course of your lifetime. That's not saying that one mutation can't cause cancer, but usually it's like an accumulation of mutations. This is why cancer is much more common in elderly patients as opposed to really young people. But like I said, I mean, you can still see cancer in children, so this isn't a steadfast rule. It's just kind of a guideline. Usually it's an accumulation of mutations that ends up leading to the development of cancer. So, just some definitions here before we wrap up this week's lectures. Stem cell. So what is a stem cell? There's a lot of ethical issues that are kind of surrounding stem cell research. So what a stem cell is, we have embryonic stem cells, we have hematopoietic stem cells, we have pluripotent, multipotent, totipotent stem cells. So there's all different levels of what stem cells are. So before you go and bash all stem cell research, you should have some foundational knowledge of what are the levels of these different types of stem cells. So we'll start at probably the most ethically questionable practice. So an embryonic stem cell. In an embryonic stem cell, you have an egg and a sperm that are fused together, and some people consider that life. As soon as you have that fertilization event, they consider that life. So this is where it becomes an ethical issue, and you can think about your own personal beliefs of how you feel about this. But the sperm and the egg come together, they unite, and they let it divide and divide into 4 cells. Now they take cell A and they ship it off to Japan, and Japan uses those cells in order to develop their own stuff. They take cell B and ship it over to Canada, and Canada uses that cell, that embryonic stem cell, in order to produce their things. And cell C and D go to different locations as well. So now people are saying, okay, well, you're taking living humans and you're spreading them all over the place, and that gets into a lot of those ethical issues. This isn't done every day. We have this subset of existing stem cell lines, and those are the lines that are functional lines that we're using. So we're not constantly producing new fertilization events to produce new embryonic stem cell lines. We're using the existing ones to do research on them. Now that's an embryonic stem cell, but once that stem cell differentiates, it can become a certain type of cell. So there's different cell lines, like a hematopoietic stem cell would only be able to become any of the blood cells. We have mesenchymal stem cells, which are a little bit less differentiated, and mesenchymal stem cells can become fat cells, bone cells, neurons, or hematopoietic stem cells. So we have all of these different layers of how differentiated they get. Once you get beyond a certain point, though, you're kind of committed to become a certain type of cell. So an embryonic stem cell can become anything in the body, while a mesenchymal stem cell can only become a bone cell, a fat cell, a neuron, or a blood cell. And while a hematopoietic stem cell can only become one of the blood cells. So as we increase the specialization of the cell, that's what cell differentiation is. So cell differentiation, we're increasing the specialization of a cell. A stem cell is an undifferentiated cell that can become any cell type. Then we have apoptosis. Apoptosis is programmed cell death. A cell gets a signal to kill itself. And in a multicellular organism, that's good. As we develop, we all have tails. In embryonic development, all developing embryos have tails. We don't want to be born with tails. So our tails get the signal, all of the cells in our tails as an embryo get the signal to self-destruct. That's apoptosis. So apoptosis is programmed cell death. If you ever have excess cells that are no longer needed in the body, why would you waste the energy to support those cells? Give them the signal to kill themselves and they'll go away, and then the multicellular organism benefits from that. We've tried to induce apoptosis in cancer cells. We try to program the cancer cells to kill themselves. And many researchers are really close to getting to that, to getting those cancer cells to just self-destruct and go away. And some have already achieved that. It's just a matter of getting the medications to the market, which we all know takes at least 10 years from research to market. But apoptosis, again, that's programmed cell death. Hypertrophy. Hypertrophy is causing an increase in cell growth. Now, hyper means more. Trophy is talking about, hypertrophy is talking about the increase in the size of the cell. So you're increasing the size of the cell. Hyperplasia is talking about increase in cell number. So it's still an increase in cell growth, but now you're increasing the number of cells, where hypertrophy is just increasing the size of the cell. Hyperplasia could indicate cancer. It could indicate a benign tumor, but it's just increasing in cell number. It could also just indicate normal function. Like you have to produce more cells in order to do a particular function. Dysplasia. So dysplasia is when you have a growth of cells into a tissue layer that it's not intended to grow into. So if you have the beginnings of cancer, usually the cells, the cancer cells, would break through the basement membrane and you'd have dysplasia happen. Neoplasia is new growth. Neoplasia could be indicative of cancer. So if those cancer cells dislodge and put themselves in a different place, then that could indicate cancer. Atrophy. Atrophy is the decrease in the size of an organ or cell due to a lack of nutrient or nerve supply. So it's a decrease in size of an organ or cell due to the lack of nutrients or lack of nerve supply. An example that I have for this one is my dad. So one of my dad's legs is like a quarter of the size of the other one of his legs. So he refuses to wear shorts because he doesn't want anyone to see his like tiny little leg. But he originally thought it was post-polio syndrome. In post-polio syndrome, you have a lack of nerve supply that can cause flaccid paralysis of one or both of your legs. Usually it's more common in the legs than in other parts of the body. But that can lead to a decrease in the size of that body part. So the legs can kind of waste away, or one of the legs can waste away. And because my dad had polio as a kid, because his dad was in the military, is from Puerto Rico, and they traveled all over the place, they never got anyone vaccinated, and he ended up contracting polio on one of my grandpa's tours in Germany. This is what he assumed that he had. So he went to the doctor, and the doctor said, well, good news, you don't have polio, but you do have a pinched nerve going to your leg, so we have to do a spinal fusion. So my dad had the spinal fusion done, and it decompressed the nerve, and he regenerated a lot of the nerve sensation to the leg, and now his leg is a little bit bigger. It's still not 100%, but some of that nerve function was restored. It was really painful while that was repairing itself, because all of a sudden having all of that nerve stimulation from parts of your body that were basically deteriorating was painful for him. But atrophy, because that nerve was pinched, he wasn't getting adequate nerve supply to that leg or to the muscles of that leg, and that decreased the size of the muscle cells in the leg. So atrophy is a decrease in size due to a lack of nutrients or nerve supply.